Good afternoon. The topic for today's lecture is going to be the topic that Darwin referred to as the mystery of mysteries, which is speciation. Speciation is basically just the formation of new species. And Darwin's big question was, how do new species form? He couldn't quite figure that out initially. So in order to understand how new species form, it is important to understand the definition of what a species is. And there are several different definitions of a species. One that most biologists focus on most of the time and use most of the time is the biological species concept developed by the evolutionary biologist Ernst Mayr in the 20th century. And he said that a species is a group of populations that can interbreed in nature and produce viable, which means living, fertile, so they can reproduce offspring. And what keeps a particular species from being able to, diverging into two new species, but in fact holds that species together, is gene flow, right? As long as there's gene flow occurring between populations of the same species, different populations of the same species, then they won't diverge from one another. But if there is something that can interrupt that gene flow and prevent two different populations of the same species from interbreeding, then those two populations may diverge from one another genetically as they as natural selection takes its course in each of their two separate populations. So in the picture here, we see that we have two birds here that look very similar, but are actually different species because they live in different areas. There's been no gene flow between these two populations of birds, and they've diverged far enough from one another genetically that they will no longer interbreed in nature. Whereas the human population, although we live all over the globe, and we have several different human populations separated from one another, there is still gene flow between all those different human populations that hold them together, and we remain one species. So in order for new species to form, something called reproductive isolation has to occur. Reproductive isolation is just basically any biological factors that impede members of a particular species from producing viable, fertile offspring. So anything that stops members of a species from producing fertile offspring with another species is a reproductive isolating mechanism. And these reproductive isolating mechanisms block or limit gene flow, and so they prevent the formation of hybrids. These isolating mechanisms are classified generally on whether they occur before fertilization or after fertilization. Any factors that occur that prevent members of two different species from producing viable, fertile offspring that arise before fertilization can occur are referred to as prezygotic barriers, and any ones that arise after are called postzygotic barriers. So we're going to take a look at each of the different types of prezygotic and postzygotic barriers. The first prezygotic barrier that can exist is what's called habitat isolation, where members of two different species occupy different habitats within the same area. So we may have two different species of garter snakes where one lives in the water, one lives on land, so they never actually encounter one another in nature, so they never interbreed, so they don't produce any offspring. So that is a prezygotic barrier. It arises before they even attempt to mate. We can have what's called temporal isolation, where species, members of different species breed at different times, either of the day or seasons or years. For example, we have the eastern and western spotted skunk. Their ranges overlap, but the eastern spotted skunk mates in the late winter, and the western spotted skunk mates in the late summer. So they are not looking for mates at the same time. So even though their ranges overlap, there is a reproductive isolating mechanism. There is a prezygotic barrier that prevents them from breeding, so there's no gene flow between them. Another prezygotic barrier is what's called behavioral isolation, where members of different species have different courtship rituals or behaviors. There are the blue-footed boobies, have a very distinct courtship behavior that if any other bird species were to observe, they would not know that was a courtship behavior. That would not draw them near. Giraffes and albatrosses as well. These are video clips that aren't going to really show up, I don't think, if I play them here. So if there's time, I will link these in the folder for today, so you can watch those different courtship behaviors if you want to see them. There's what's called mechanical isolation, where basically the reproductive parts don't fit, so any mating attempts between different species are unsuccessful due to morphological differences, morphological meaning form differences. For example, bush babies. There are several different species of bush babies, these little cute little looking mammals right here, but they have unique genitalia where the male and female genitalia fit together like a lock and a key. These are the various shapes of the penis of a bush baby, and you can see how these would be unique to each particular species and only fit a female of their particular species. All right, and then finally, the final prezygotic isolating mechanism, or is referred to as gametic isolation, and this is where the sperm is unable to fertilize the egg. So the members of the different species do try to come together to mate, but the sperm is unable to fertilize the egg, and that's usually due to chemical signaling. For example, in sea urchins, that we have what are called red sea urchins and purple sea urchins, and they will release their egg and sperm into the water near each other, but they can't bind. The sperm are not drawn to the egg. There's not the right chemical signaling to draw the sperm to the egg of the different species, and so again, they remain separate species because there will be no gene flow between them. Okay. So those are the 5 prezygotic isolating mechanisms that prevent gene flow between 2 different species and help to maintain them as separate species. We can also have what are called postzygotic barriers. So members of different species do attempt to mate, and they do form a zygote, but still there will end up not being successful gene flow between the 2 different species. The first postzygotic barrier is referred to as reduced hybrid viability. So the hybrid forms, but it does not complete its development, or it's really frail. For example, the salamander species Ensatina, the hybrid that forms between different species of the Ensatina salamander are very, very frail and don't survive to reproduce. We can have what's called reduced hybrid fertility, where the hybrid is vigorous, but it is sterile. So that's usually because the 2 parent species differ in chromosome number or structure. For example, you probably know the very famous example of a horse and a donkey producing a mule. Mules are bigger than a horse or a donkey. They're very strong, but they are infertile. So they are not, again, a horse and a donkey are not going to be able to ever have gene flow between them because they produce this sterile offspring. And then the third type is referred to as hybrid breakdown. With hybrid breakdown, what happens, the members of separate species breed together. The first generation is actually of offspring, the first generation of hybrid offspring, it's actually viable and fertile, but the offspring of the next generation are feeble or sterile, so they can't continue on. And this happens with some strains of cultivated rice that we see. All right, so now what we've got here is that there are some problems with the biological species concept. There are some limitations to it. So I'm going to pause real quick and let you think, what are some reasons why biological species concept isn't always a great way to classify, right? So if you think about it, there's got to be a couple things that hopefully you could come up with, right? First of all, you can't apply the biological species concept to things that are no longer living, right? You can't apply the biological species concept to fossils. We can't tell whether 2 fossilized extinct organisms would have been able to reproduce and produce fertile offspring. There's just no way to know. it can't be applied to asexually reproducing organisms, right, that don't mate with another organism. And it is defined by the absence of gene flow. So in nature, sometimes we see morphologically distinct or ecologically distinct, so they play different roles in their environment if they're ecologically distinct. Morphologically distinct, meaning they don't even look close alike. They can still have gene flow. So there are a few other definitions of species that are sometimes used. One is called the morphological species concept, where different species are classified, or organisms are classified into different species based on their appearance. There's the ecological species concept, where organisms are classified based into different species based on the role that they play in their environment. And then there's what's called the phylogenetic species concept, where organisms are classified based on their evolutionary history and placed into the smallest possible groups of organisms with a common ancestor. So there are some other ways to classify organisms that don't fit the biological species concept for one reason or another. All right, so how does speciation occur, right? Well, it can occur if organisms become geographically isolated from one another, but they don't have to become geographically isolated from one another in order for new species to form. So we'll take a look. In what's called allopatric speciation, gene flow between two separate populations gets interrupted when they become geographically isolated. So if a canyon forms, or if a river changes course, like the Mississippi River changed course when there was a huge earthquake I think it was the 1800s. It changed the course of the Mississippi River because of the earthquake. And so now organisms that had been all living together as one population end up being split into two separate populations on either side of a canyon or on either side of a river. And so their environmental selective pressures on either side of that physical barrier can be different. And so because of that, different selective pressures will favor different adaptations, and you can end up with different species evolving over the course of time because of natural selection acting on those two separate populations. You could also have a mountain range form to separate populations as well. So here we have ground squirrels, two separate species of ground squirrels that you see here in the picture. That were separated when the Grand Canyon formed. These used to belong to the same population of ground squirrels. Grand Canyon formed, they are separated from one another. The ground squirrel can't quite jump across the Grand Canyon, and so on either side of the canyon are different selective pressures, leading to the accumulation of different adaptations in the two populations. Eventually, they diverged enough that they became separate species. So the process of allopatric speciation is pretty simple, right? You get the separation, the physical separation of populations, followed by natural selection acting on both populations. So you can read in the book about the evidence of allopatric speciation on your own. I've given you a couple of examples here. The type of speciation that I think is a little more difficult to understand is what's called sympatric speciation, where you get speciation occurring, you get gene flow interrupted between populations of organisms that live in the same area. What could it be? If they're not physically separated from one another, why would gene flow stop between these populations? Okay, so this shows you here in allopatric speciation, you had a physical barrier formed between what used to be a single population of fish. A physical barrier formed, separating them, so you had different selective pressures in the two pools of water, selecting for different adaptations, and they became separate species. But in sympatric speciation, no physical barrier arises between them, yet we end up with a new species forming here. So how does that occur, right? Well, it can occur via what's called polyploidy, which is errors in cell division that lead to extra sets of chromosomes. This doesn't happen frequently in animals. I believe it's the gray tree frog it's happened in, but it happens pretty frequently in plants. Okay, in plants, you can have what's called the formation of an autopolyploid. which is an individual with more than 2 sets of chromosomes, and that's derived from a single species. So here was the original diploid cell of a particular plant species, typically. Then you get the failure of cell division after duplication. So you get tetraploid tissue here, right? So any gametes that are formed after this failure of cell division, any gametes that are formed are going to be diploid. And so if they were to, say, fertilize through self-pollination like some plants do, or with another member of the same population that had the same type of polyploid event occur, the offspring are going to be tetraploid. And they can be viable and fertile. Now, one thing to think about, right? This particular plant that produces these diploid gametes would not be able to produce fertile offspring with a plant that hadn't gone through this polyploidy event right here, the duplication of chromosomes, because any plant that is diploid will produce a haploid gamete. So if you have a diploid gamete fertilize a haploid gamete, you end up with triploid. You end up with 3 sets of chromosomes that cannot be divided equally up into gametes. So then you end up with a sterile offspring. So this particular plant would be a different species than the original parent plant that it formed from. All right, now in case you think I'm crazy in showing you these polyploid events, like what do you mean duplication of a whole set of chromosomes? How is that even possible? That seems like no way. Well, I was doing some research and I found this in a Nature journal from 2009, June 3rd of 2009. It says that polyploidy is a major creative force in plant evolution from ferns to flowering plants. Estimates suggest that approximately 70% of flowering plants have undergone at least one whole genome duplication event during their evolutionary history, and that 2 to 4% of all speciation events involve polyploidization, so the polyploidy occurring. So this is a significant event in the evolutionary history of plants. It does happen frequently, it does happen often, so I'm not just making this stuff up. Okay, so in allopolyploidy, right, what we just looked at was autopolyploidy, where you had the autopolyploid error in division of the chromosomes, and then you get self-pollination leading to the tetraploid offspring. In allopolyploidy, you end up with this occurs between members of 2 different species here. So it wasn't within the same plant, it wasn't within the same species. So species B here has a diploid number of 4, species A has a diploid number of 3. So if this species B goes through that polyploidy event where it produces gametes with an unreduced number of chromosomes due to failure of those chromosomes to separate during meiosis, you would have a gamete with 4 chromosomes here, a gamete with 3 chromosomes formed through normal meiosis from species A. And so what's going to happen? They produce if fertilization were to occur between these 2 gametes, they're going to produce a hybrid with 7 chromosomes. Now that hybrid itself is going to be sterile. It's got an odd number of chromosomes. However, if it produces gametes, unreduced gametes with 7 chromosomes. So if it goes through meiosis, and again, we have this polyploid event occur, it can produce gametes with 7 chromosomes. The chromosomes didn't separate. And then species A here goes through normal meiosis and produces a normal gamete with 3 chromosomes. Fertilization event occurs again. We end up with an offspring with 10 chromosomes, a diploid number of 10 chromosomes. Those chromosomes can be divided up equally, and we now have a fertile hybrid offspring. This seems bizarre, I know, but it happens, right? And this is actually been documented as happening in plants, not necessarily in animals. All right, another way that Sympatric speciation can occur is when we have what's called habitat differentiation. So when a subpopulation of a particular species exploits a habitat or resource not being used by the parent population. The North American maggot fly is an example of this. So the parent population of the North American maggot fly lived in hawthorn trees. Some individuals from those hawthorn trees ended up colonizing nearby apple trees. Now, what's interesting to note, the food source, if you were a maggot fly living in an apple tree, is going to be the apple fruit. And the apples matured more quickly than the fruits of the hawthorn tree. So what happened was the selective pressure was the North American maggot flies that were populating the apple trees, the ones that matured more quickly and could take advantage of the food source when it was available, had a selective advantage, survived better, reproduced more often. And what happened over the course of time was that the populations that live in the apple trees develop more rapidly than the ones that do in the hawthorn trees. And that eventually led to the temporal isolation of the two different populations, because the ones in the apple tree are developing more rapidly and maturing more rapidly. They reproduce earlier than the ones in the hawthorn trees do. So they're temporally isolated from one another. Sexual selection can also drive sympatric speciation. In Lake Victoria, there are 600 species of cichlids. That's a type of fish that formed from a small number of colonist species. And the females of a particular species select males due to their breeding coloration. And so the different breeding colorations of the males is what keeps the species as being separate populations. In fact, when an experiment was done where members of different species of cichlids were put together in a tank with the same color light shining on them, so that their coloration all looked the same, females could not choose or did not select males of their own species necessarily, and were able to produce offspring. But because in nature they don't, they're still considered separate species. But the fact that they can still produce viable, fertile offspring tells us that the speciation event didn't occur that long ago. But again, they don't do that in nature. They don't breed together in nature, so they are separate species. And actually, there's been a decrease in the number of cichlid species in Lake Victoria. Some of that is attributed to the introduction of a non-native predator fish called the Nile perch into Lake Victoria, but also the water in Lake Victoria is becoming more and more polluted, so it becomes more difficult for the females to distinguish the males based on their coloration because the water is more polluted. So we're seeing a decrease in the number of cichlid species in Lake Victoria. So, you know, that mystery of mysteries, how species form, that question has kind of been answered, right? We understand how speciation occurs now. Speciation is referred to as macroevolution. We kind of understand how that happens now, right? Through reproductive isolation, reproductive isolating barriers that arise. But another question that biologists ask is, what is the time course of speciation? Is it quick? Is it slow? How does it work, right? How quickly does it occur? And it turns out that it can occur relatively quickly. Stephen Jay Gould and colleagues at Harvard proposed the concept of punctuated equilibrium, where what the time course is that new species appear suddenly and then stay the same for long periods of time, and then there are rapid bursts of change again. Now, that rapid amount of time evolutionarily could be 5,000 years. When you consider that the Earth is over 4 billion years old, 5,000 years is a really short period of time. But with some other species, we see a slow transition from ancestral species to modern species, where changes take millions and millions of years, right? So Stephen Jay Gould's proposal, where organisms appear suddenly, that occurs sometimes, that's referred to as punctuated equilibrium. In other cases, for other organisms, there are very slow changes that occur gradually over millions of years, and that is referred to as gradualism. So that is the, speciation, once it begins, it can occur rapidly, but it could also occur slowly. And so that is the end of the presentation. I hope you guys are all happy and healthy. And I will see you guys sometime soon, I hope.